We are here at Garrett Morgan School of Engineering and Innovation working with our students. Our volunteers are here every Wednesday in the spring. What happens if a seed, like a plant seed, is ingested? Does it actually germinate better if it's actually gone through the digestive tract? So they're feeding earthworms different nitrogen-rich seeds that they research to find out the best ones to use, and they're seeing whether or not the plants grow quicker.
The first seeds we tried, the worms didn't eat. So, I guess we just got the worms that the seeds that the worms were actually eating.
Nice.
You see, like, green string.
Oh, yeah.
Did you guys have anything happen that you weren't expecting during the project?
Yes. Yes. So the worms escaped and they died. Yeah.
It was so big.
We don't even know how, because they were in a box. And then they
escaped outside the box?
They were in one of these, and then we put them in the box, and then they escaped. I don't know how.
I know you had this idea about if somebody dies, they could eat a seed before, when they're in the hospital, and then a tree could bloom or something. What type of plant do you want to eat on your deathbed?
Sunflower.
All right, so we're here in our second group at Garrett Morgan, and they are using C. elegans, a type of nematode, to understand more about gene-drug interactions. Yvonne, what type of animal are you using?
It's called worms.
The worms?
Yeah.
Awesome, very cool. And what is it that you're hoping to see?
How the CBD affects them in their movement.
Cool. So you're feeding them CBD and then seeing how their movement changes. That's awesome. All right, Daniel. So what has happened so far that you weren't expecting?
When we were doing it, we put too much CBD and the worms started to die because they were eating so much of it. Oh my goodness. So that was unexpected.
Carter and Logan, what has been your favorite part about this so far?
I think using the microscope here,
yeah,
it's got to be the best microscope.
The microscope. So in this group, the students are interested in seasonal depression, which is really, really important, especially here in Cleveland, Ohio, where we have to worry about cloud cover in the winter. All right, Naisha, so what type of animal are you guys working with?
Fruit flies.
What have you thought so far about working with fruit flies?
They reproduce a lot.
Favorite part so far of this project?
Sorting them out, because you get to see up close how they look under the microscope.
Is this what you thought science was going to be like?
No.
No. What is different than what you expected?
I thought it was gonna be like with potions, like glass and stuff like that.
Oh, yeah. Keep tapping. Light tapping. Keep tapping. Lift.
Y'all put it on the wrong thing.
All right, so we're here now with this group who is studying how ingesting components of marijuana like CBD can change your appetite. Kiana-Lee, so what is it that you're working on?
We just transfer the one that has CBD in the food container.
Whoa, that's cool.
There are a lot.
So those flies have been exposed to CBD?
Yeah.
Wow. So Xavier, what do you think is going to happen between the flies that have CBD and the flies that have no CBD?
I feel like the flies that got CBD gonna eat the food faster than the flies without it.
That's really cool. And you guys, you mentioned that there's different foods. So you said fructose, sugar, and quinine, and olive oil. So what is the difference between those? Why did you guys decide to use those? Jovan, do you mind sharing a little more about the different types of foods that they're eating?
I think it's olive oil, some quinine, something like that.
Yeah.
Yeah.
And the quinine, that's bitter and gross, right? Do you guys, what do you think? Do you think the CBD flies are going to eat it anyways?
Yeah.
You think that's going to be it's like eating old Taco Bell the next morning? They just don't care, right? They don't care. They got the munchies. They're going to eat whatever they have. Can you walk us through what you did to make the tubes?
So we cut up a sponge to block up the top, and then we put the pipettes, and then we stuck them into the sponge, and then we basically wrapped it up so that we could see how much food they're eating.
That's really cool. So you're doing some engineering too, along with your biology. We're in Miss Herman's class now, and we're going to hear from 4 student groups here to hear about the groundbreaking discoveries they're making. All right, so we're at this group that's interested in a really important question about generational famine and how having periods of famine might affect the offspring. So Jamali and Michael, they are going to tell us a little bit about their project. What type of animal are you using to look at this?
Fruit flies.
What has it been like working with fruit flies?
Highly disgusting.
Good, you say highly disgusting?
Yes.
Yeah, what has been gross about it?
Mainly the maggots and being freaked out about them coming awake when we were putting them to sleep.
Yeah, because you guys anesthetize them, right? And sometimes they'll kind of wake up a little. Awesome. And today, what are you guys going to be doing?
Dissecting them, their brains.
What made you guys interested in this question?
I was doing research because I didn't really know what I wanted to do at first. And then it popped up and I was like, oh, I really want to learn about that, you know?
We're moving to a group that is trying to understand more about addiction and caffeine consumption, which I think is really interesting because I drink a lot of coffee. So let's see what they found out. What type of animal are you guys working with to study caffeine?
We're working with earthworms.
Earthworms, very cool. Have you guys found anything surprising?
The worms that have more caffeine are like sluggish and they just seem to be struggling with most of the tests. But the worms that don't have any caffeine are just regular.
That's a little unsettling. Has this changed your behaviors at all outside of the lab?
Like the amounts of caffeine like I drink way less now.
And I think I recall when you guys first started, something crazy happened with like the highest amount of caffeine, right?
Yeah, they all like kind of shriveled up. They lost a lot of their mass, and we dissected them afterwards.
Like it was like gross for me to clean them because they were already just like really skinny and it was gross.
Yeah, like
So each container of soil has different levels of caffeine. So yeah, our control obviously has none, and like it gradually gets higher. The caffeine was in pills, so we had to break like open the capsules and then pour it into a beaker and then put water in it to mix it.
All right, so now we're going to talk to this group who's interested in mechanisms of antibiotic resistance. Let's hear what they find. What are you guys working on? Can you tell us a little bit about your project?
We're making like a superbug basically. So it's like bacteria that's like resistant to like different antibiotics.
Have you guys been like a little nervous doing this? I see you're all wearing gloves and following safety protocols, so that's great.
Pretty good. We've been wiping everything down. I mean, I don't really think there's too much to worry about.
So what made you guys interested in this question?
Should really like to see like the progress and like all the steps it takes to like actually find what we were looking for.
That's really neat. So far in your project, what have you learned that you weren't expecting?
I mean, like we went a lot of trial and error, like for some of them we used too many antibiotics and like we didn't get the results we wanted, so it's like we just had to kind of keep redoing it with different like steps to see the different results.
That's very much what it's like in the lab. We do a lot of revising and trying over and over again.
Like seeing observing how bacteria can affect how how bacteria can evolve, you know? Just seeing how something could evolve in such a short period of time just makes us wonder how humans could evolve in the future.
I don't know, I think it's like opens a lot of opportunities. Like I have like experience if I ever want to do something like this in the future.
Has this been what you expect for science? Is this something that you think is fun?
Superbug
makes me smarter.
It's a great opportunity to like learn how to do things that you're actually interested in. Like this was like my idea and like we're doing it.
Awesome.
So I think that's like really cool.
Yeah, for sure.
I think it's really cool and it makes me wonder about how the cells in our bodies work.
So now we're going to talk to one of our students representing a group that's looking at how diet can actually exacerbate symptoms of Alzheimer's disease using fruit fly genetic models, transgenic models of Alzheimer's disease. What is it that you're working on?
Taking this fly's head off so we can see the motor skills, I think.
Right now you're practicing on flies, but you have some flies right now that are developing Alzheimer's disease. What type of things are you changing in their diet to see, and what do you expect that's going to change?
I'd say they forget where they are probably if we change the food we give them or how much we give them.
What makes you interested in Alzheimer's disease? What made you curious about this question?
Alzheimer's definitely interested me because I think you can get your memory back if it's either Alzheimer's or dementia, because I heard you just lose access to your memory rather than actually losing it.
What's been your favorite part about the project so far?
No work.
Yeah, being able to do some hands-on stuff. So this doesn't feel like work, even though you're doing like cutting-edge research. All right, we're here with Ms. Herman, talking to her about her experience with Rise Up. What has it been like so far?
Oh, it's been awesome for the kids to actually get to be involved in the entire scientific process, actually doing research methods and applying the different things that we learn about in biology to actual experiments and things that come from their own ideas and that they actually have interest in.
Our students that are at different schools, so we're going to be showing them some of this content in these videos. Is there anything you want to let the other students know that are at different schools about your students or about the program or your experience?
I would just say, it's been a great experience. I hope it's been as awesome for them as it's been for us. I'm really interested to hear what some of their projects were as well, and kind of just see what they've been up to at the other buildings. You guys send out the newsletter, which is really cool to see what's been going on, but really to just kind of see what their results look like too. I think will be really cool, and for everyone to get to be published is going to be awesome.
Thank you so much for letting us come into your classroom and sharing your students with us. It's been really, really rewarding.